Hi everyone, and welcome to the NanoCellect presentation about the benefits of cell sorting prior to genomic application workflows. We are pleased to participate in the Genetics Virtual Week, and today we have two speakers for you. First, we'll hear from Emily Bozek, Product Manager here at NanoCellect. Emily will discuss why low-pressure cell sorting can reduce noise in sequencing data and how it ensures an end-to-end successful 3' RNA-seq workflow. Next, we'll hear from Dr. Nicole Jagnandan, Application Scientist at NanoCellect. Nicole will highlight how microfluidic cell sorting compares to traditional cell sorting for sample preparation prior to 10x Genomics applications. If you have any questions, feel free to submit them through the Labroots platform, and we'll be sure to get back to you.
Good morning, everyone. My name is Emily Bozek, and I am the Product Manager for the Wolf at NanoCellect Biomedical. Today, I'll be covering high viability cell sorting for single-cell 3' RNA-seq. So the agenda for today, I would like to cover some issues that users may run into when they're completing a 3' RNA-seq protocol. I'd like to review why low-pressure cell sorting can reduce noise in sequencing data. I'd also like to cover how to complete a full end-to-end workflow for a successful 3' RNA-seq experiment with cell sorting as a sample prep stage. And then at the very end, I'd like to go over a case study from a customer site at UCSD, which is in partnership with the Shiley Eye Institute around a neuronal gene expression study. So first of all, why would researchers want to use 3' RNA-seq as opposed to other RNA-seq methods? So for some people, it's important to understand that for traditional RNA-seq, it generates sequencing libraries from whole transcripts, like this top example shown here, as opposed to 3' RNA-seq, which only generates a single initial library molecule per transcript, which is complementary to 3' end sequences, shown in this bottom example. One limitation of the more widely used longer transcript RNA-seq method is that those longer transcripts are represented by more reads than the shorter transcripts, which may result in a biased estimation of expression levels. So 3' RNA-seq would suggest that it has a greater power to detect expression variation using that particular method. Other advantages to 3' RNA-seq are that it typically costs half or less than that compared to some of those other single-cell methods. In general, it has a simpler library prep protocol, so kind of less hands-on activities to do. And because it uses fewer reads than traditional RNA-seq, it tends to be more suitable for labs that may have, smaller NGS instruments or potentially larger sample sets. So kind of amending that protocol for those particular use cases. And then again, just to kind of reiterate, 3' RNA-seq is typically most useful to figure out differential gene expression from different sample sets. So typically when users are going through RNA-seq, and then also for 3' RNA-seq, they like to use FACS technology, so fluorescent activated cell sorting technology, to dispense a particular population of cells using this flow cytometry method to dispense into single wells of a PCR plate. Now, the issue is that traditional FACS, for a number of different reasons, isn't typically well suited for a number of genomics assays, including 3' RNA-seq. So some of those issues, number one, are just around time and being able to schedule using some of these complex instruments as a part of a core facility at a university or some other institute. It can just be really kind of cumbersome to have to travel to that core facility, depending on how large campus is, schedule that time so that it works with your particular sample type, which may be coming from in vivo samples, and really just not having that convenience of being able to kind of work around your schedule and having to work with someone else's. There's also a higher risk potentially of contaminating your samples from previous samples carryover because these traditional instruments don't have disposable fluidics. So those lines from the fluidics cart traveling into the machine typically are not replaced that often and can be subject to microbial contamination over the course of a period of time. Now, they do have cleaning methods, but that doesn't always guarantee that it's that you're completely getting rid of free nucleic acids or free RNA that could be floating around and then potentially contaminating your sample and then therefore contaminating your sequencing data set. It can also just be simply quite expensive to optimize if you're starting a new assay, a new sequencing type of assay, and have to figure out some of the correct settings or cell populations that you want to probe deeper into. Just the cost around scheduling that time and that expert availability just can be quite expensive. And then finally, probably maybe perhaps the most important for some genomics experiments is that these traditional sorters, the jet, nair, and cuvette-based ones, can induce a lot of cell stress by high pressure, decompression shock, and then electrostatic charge on those cells that are getting dispensed into plates. So all these things, by inducing cell stress from all those factors, can have an effect on gene expression and potentially cloud the types of conclusions that you would come to from that type of data. So, just inducing more error than needed potentially. Now, when you're doing cell sorting for genomics experiments, it's very important to get rid of dead cells, debris, and doublets. And even with 3' RNA-seq, you definitely want to have very good sample prep upstream of this. It's quite difficult for a lot of different sample types to have a high degree of dead cells and debris. So for this particular sample, you're seeing here in red on a 2D scatter plot, forward scatter versus side scatter, a high degree of dead cells and debris shown here in red. This particular sample was stained with PI. And then you can view that with fluorescence with the right side of this histogram plot over here. So there's a very high degree of PI positive or dead cells contaminating this sample, which would really have users potentially having a fair amount of free RNA or free nucleic acids in their sample, which could, kind of cloud and, make the data set a lot more noisy than it needs to be. Other users will also find potentially that their samples are quite sticky and can have a high degree of doublets. So doublets can induce false positives and kind of cloud sequencing data a little bit more. So doublets typically in flow cytometry plots show up over here and can just make the data a little bit noisier also than it needs to be. So all of this with traditional sorters, knowing that because of that high pressure and shock decompression, you can really get a lot of partially degraded total RNA or many peaks of completely degraded total RNA, which really will have an effect on the quality of your sequencing data set. So a solution to all of these issues that I mentioned, if you're trying to go through 3' RNA-seq and then also use a type of flow cytometry-based cell sorting system, would be to use microfluidics. So the Wolf benchtop sorter and N1 single cell dispenser from NanoCellect allows users to do a very simple cell sorting single cell dispensing into PCR plates at very low pressure, so less than 2 psi, no decompression shock, and of course no electrostatic charge. So the Wolf in combination with the N1 single cell dispenser, so the Wolf with the N1 single cell dispenser will gently sort a cell type of your choosing into PCR plates directly upstream of those 3' RNA-seq kits. So to validate that the Wolf and N1 system does actually give you, good single-cell efficiency, we did a number of experiments comparing single-cell efficiency from flat-bottom culture plates versus the more conical bottom PCR plates. What we did here, it was an experiment with beads where for the culture plates, we did a full 8 plates of 96-well dispensing versus 13 plates of 96-well PCR plates. We did not find any significant difference between those single-cell sorting efficiencies between those two different plate types. These cell culture plates on average had 98% single-cell efficiency, while on average the PCR plates showed 90% single-cell efficiency. So in order to fully validate that the Wolf and N1 system does work well for 3' RNA-seq, we did a few experiments by using the QIAGEN UPX 3' transcriptome kit. So 2 plates of 293 T cells were dispensed using the N1, and then library prep was completed using that QIAseq 3' transcriptome kit. cDNA was sequenced and analyzed, and the estimated cell number per well was defined as the following criteria. So if it was less than 100 genes, that was relegated to being 0 cells. If it was 100 to 1,499 genes, that was determined to be 1 cell per well, and anything above 1,500 genes was 2 or more cells per well. So we estimated for 1 plate, based on this analysis, that about 72% of one of the 96 well plates had 1 cell per well, and then 92% of the second plate had 1 cell per well. So these results just show that the Wolf in conjunction with the N1 are able to successfully plate a single cell into a PCR plate and then actually give you relevant results for 3' RNA sequencing. With that, we do present a full end-to-end workflow using the Wolf cell sorter and the QIAseq UPX 3' transcriptome kit for 3' RNA-seq. So all users have to do is begin with a mixture of suspended cells, load it onto the Wolf cell sorter. You will figure out which population of cells you're actually trying to sort, and then you'll dispense it using the N1 single cell dispenser, dispense those single cells into 96-well QIAseq plates, which are preloaded with a cell lysis buffer. So those cells then lyse, and single-cell RNA can then be reverse transcribed, and each RNA molecule is then given a unique molecular index, or UMI. With that, they're also assigned well-specific cell IDs, up to 384 wells. And then following reverse transcription, with integrated template switching, all those cDNAs can be combined into one tube, enabling kind of a full library construction. So the Wolf and this 3' kit really allows you to go through a high throughput next generation sequencing workflow using polyadenylated RNAs and then sequencing using Illumina sequencer. The way that QIAseq UPX 2' transcriptome kit works is that every one of these wells in the plate has that unique primer, UMI, and cell ID mix into each well, so you really can pool all of those wells that had a single cell in it into one tube for library construction. And then finally, with the cell analysis that you would do after sequencing all of that, depending on the reads that you have, you would just need to understand the original ratio compared to the experimental ratio to be able to determine how many UMIs there were per that gene, per the sample set. So that allows users to truly kind of understand at the end of their experiment how they can compare UMI reads to the beginning mRNA or the number of genes that they originally had in that sample. So finally, the case study that I'd like to highlight with one of our customers at UCSD, working with the Shiley Eye Institute, was around a specific set of experiments related to glaucoma and then eventually other eye-related diseases like macular degeneration. So this particular lab had two different populations of mice, so wild-type mice and then mutant mice. The mutant mice groups have a particular variant that is believed to be protective against glaucoma. So by using control neurons for each of those groups and then treating those eyes with IOP, so intraocular pressure, you can isolate those particular neurons from those retinas and then sort them on the wolf via CD90.2 gating. So these four different conditions, so the wild-type mouse, control versus IOP, and then mutant mouse, control versus IOP. These four different sets of neurons then were sorted on the wolf via CD90.2 FITC gating. So this was just a typical kind of immunostating protocol where he essentially just sorted based on positive CD90.2 FITC signal and then plated into those Chi-seq plates, so single cell plating into Chi-seq plates, and then completing the library construction, NGS sequencing, and then finally data analysis. So coming out of that, the group was able to find via this TISNE analysis that each of those four different groups of neurons actually did have different types of gene expression, and you can see those clusters based on those four different conditions over here. And then finally, looking at the top 145 genes via a hierarchical clustering of those gene expression levels, you can actually see, really different expression based on the condition of the types of neurons that different genes really are expressed in different levels based on, where they came from. So, knowing all of that, the molecular mechanisms of glaucoma and then some of these other eye-related diseases that this group is working on will be able to be, begin to be elucidated using this particular method. So just in summary, we were able to show that high quality sample prep using the Wolf and N1 cell sorting system was pretty key to generating good quality sequencing data. The 3' Chi-seq UPX kits from QIAGEN are compatible with the Wolf cell sorting system. And then that Chi-seq kit and the Wolf are accessible tools for a real-life case study for researchers to study single-cell gene expression in neuronal cell populations.
Hello, thanks for joining me today. My name is Nicole Jagnandan, and I'm an application scientist at NanoCellect Biomedical. I will be discussing how you can benefit from using a cell sorter prior to your 10x Genomics experiments. During this presentation, I will be talking about the issues users may run into when not taking the necessary sample preparation steps for a 10x Genomics experiment, how low-pressure cell sorting can reduce background noise and improve your sequencing results, how cell sorting fits into a 10x Genomics workflow, and then finally, I will be going over the results from two 10x experiments we did, comparing unsorted versus sorted PBMCs, and another 10x experiment with stem cells using the Wolf cell sorter and the BD FAXARIA 2 for sample prep. When preparing your sample for single-cell sequencing, it is important that you treat your cells as gently as possible. Things such as using wide-bore pipette tips, avoiding excessive vortexing, using gentle centrifuge speeds, and using the optimal buffer for your cell type are all important techniques you should use. You should also remove any debris, dead cells, and cellular aggregates from your sample. Not taking these precautions can lead to a clog during your gem generation step, degraded RNA, poor library generation, inaccurate sequencing results, and a significant loss in money. Fluorescent activated cell sorters can be a valuable tool in your sample prep workflow for 10x Genomics applications. Debris, dead cells, and sticky cells or doublets can all be removed with a cell sorter. In addition, if you're only interested in a specific cell type from a mixed population, you can tag these cells with fluorescent antibodies to isolate your targeted cell type. However, high-pressure cell sorters can cause shear stress on cells that can lead to cell damage and gene expression changes that can alter your sequencing results. In addition, because the fluidic system in these traditional cell sorters are fixed into their machines and not disposable, you run the risk of cross-contamination between your samples, which could also lead to inaccurate results. Conventional cell sorters that use the cuvette or jet and air sorting mechanism can exert anywhere from 20 to 70 pounds per square inch of pressure on your cells, which also causes this decompression shock once they exit out of the nozzle. This can cause a lot of shear stress on your cells. The WOOF, however, emits just under 2 pounds per square inch of pressure on your cells, and therefore there's no decompression shock once they exit out of the sorting cartridge. And so cells end up being much healthier and functional for your downstream assays. As I mentioned before, cell sorters can be used to remove dead cells, debris, and doublets from your samples. Here's an example using CHO cells, where the starting population of dead cells, shown here in red, was 60%. After sorting, only 6% of the population was shown to be dead cells. Using a height versus width or area plot on a cell sorter can also allow you to identify and remove doublets from your sample. Now, if you're only interested in a specific cell type, you can also isolate that too by using fluorescently tagged antibodies while removing dead cells and doublets at the same time. Here's an example where we wanted to isolate the live T cells from a PBMC population. Our starting population had 55% live cells, which we were able to identify using a viability dye, and 21% of the cells were identified as T cells using the CD3 Alexaflor 48 marker. After sorting for the live CD3 positive T cells, we were able to increase this live population to 92% and purify the CD3 positive population to 98%. And so the main takeaway from this slide is that your sorted population is going to be a much healthier population of cells for your single-cell genomic library prep, which means you're going to have higher RNA quality and therefore better sequencing results. Another advantage to using the WOOF cell sorter versus conventional cell sorters is that the WOOF uses disposable sterile cartridges. This means that you don't have to worry about there being any cross-contamination between your samples. Everything that your sample and sheath touch go through this disposable cartridge so that you can simply switch out your cartridge between samples. This feature, along with the capability of the WOOF to be used in a biosafety cabinet, allow you to truly work in sterile conditions when preparing your sample for single-cell library generation. The WOOF cell sorter can be used prior to loading your sample into the Chromium controller. During this step, you would isolate your target cells and remove any dead cells or aggregates. After sorting your cells, if necessary, you would concentrate your sample down to the 10x Chromium kit recommendations prior to loading your sample into the Chromium controller. From there, you would proceed with their library generation protocol, and then you would sequence your samples. In order to validate the genomic benefits of using a gentle cell sorter prior to loading your sample into the Chromium controller, PBMCs were isolated from human blood and then split into two samples. In one sample, 10,000 cells were spun down and then directly loaded into the Chromium system to serve as our unsorted sample. We then took the remaining PBMCs and stained them with a viability dye, and then ran these samples through the WIF cell sorter. We then sorted for the viable PBMC population by gating out debris and dead cells using a viability dye and doublets. We then took 10,000 of these WIF sorted cells and then loaded these into the Chromium. This was our sorted sample. Both of these samples were then sequenced through the Illumina HiSeq 4000 and then ran through the Cell Ranger pipeline with the help of the UCSD Genomics core. The Cell Ranger summary produces several key metrics about the RNA and sequencing quality. The sequencing analysis for our two samples showed that there was almost 3 times more median genes per cell in our sorted sample compared to our unsorted sample. There was also more total genes detected in our sorted sample. Both of these results show a higher library complexity in our WIF sorted sample. There was also a lower percentage of reads that were not associated with a cell in our sorted sample. During the single-cell partitioning step, each cell is tagged with a specific barcode, so reads that don't have a barcode are not associated with a cell. And so this result means that there was less ambient RNA, likely from dead cells, in our sorted samples. We then looked at the t-SNE plots generated from these two samples. These t-SNE plots here are showing the heat map of the number of unique molecular identifiers or RNA transcripts in each cell. Blue meaning a lower amount of UMI counts was detected per cell, and red meaning a higher amount of UMI counts. You can see that there are much more blue dots in the unsorted PBMCs, meaning that there was much less RNA transcripts per cell compared to the sorted PBMCs. In fact, there was almost 4 times more median UMI counts per cell. This dramatic increase in UMI counts is likely due to the fact that there was less dead cells in the sorted sample. Dead cells tend to have less RNA and more fragmented RNA. There was also 2 more clusters of cells detected in the sorted sample, and the clusters and transcriptional profiles between each cell type were more distinct because of the higher library complexity and reduced background noise from sorting. We then wanted to see how these results compare to other cell sorters, so we took some induced pluripotent stem cells and split them into 3 samples. One sample served as our unsorted control, and the other 2 samples were stained with a viability dye and either sorted through the BD FACS-ARIA2 or the WIF. Both sorted samples were sorted at the same time to exclude dead cells and doublets. The ARIA sorted sample was sorted by the flow core at Sanford Burnham Previs using the 100 micron nozzle and at 20 psi, compared to the 50 micron channel in the WIF cartridge at 2 psi. 50,000 live stem cells were sorted on both machines, and then around 10,000 cells were loaded into the Chromium controller for library prep and sequencing. All 3 of these samples had cDNA libraries generated using the Chromium NextGem single-cell 3' library kit, and sequencing was done with the Illumina NovaSeq. Data analysis for these samples was also run through the Cell Ranger pipeline. The sample quality metrics for these samples showed that there was a 3-fold increase in the WIF sample and a 2-fold increase in the FACS-ARIA2 sample in the number of median genes per cell, which again shows that sorting results in an increase in library complexity. We also saw an increase in the number of median UMI counts per cell, with the highest increase in the WIF sample. Sorting also resulted in an increase in the percentage of reads that map to the transcriptome. Having a lower percentage of reads that map to the transcriptome indicates fragmented RNA or contamination. So this result shows that there was an improvement in the RNA quality and less contamination in the sorted samples. Overall, this simple experiment shows that sorting enables you to get more data per cell, and this may be even more improved by sorting your samples with a gentle cell sorter, such as the WOOF. In conclusion, the quality of your sequencing results is directly dependent on how you prepare your sample. The WOOF cell sorter is a 10x Genomics compatible product for sample preparation, enabling more data per dollar spent on genomics reagents. And using the WOOF cell sorter for your 10x Genomics applications can result in improved library complexity and less background noise from dead cells and debris, while eliminating the risk for cross-contamination. And that concludes my talk for today. If you want to learn more about these two experiments that I talked about, please visit the scientific section on our website.